Okay, so good afternoon, everybody, and very welcome to the second edition of the Sip of Science webinar series. And we have a very precious guest today, Professor Claudio Ronco. I call him the godfather of CRRT, and I just can't list the number of publications and all his contribution in the field. And he kindly agreed to give us a lecture on extracorporeal therapies, basically from diffusion to adsorption therapies. So blood purification is going to be the topic today. We're going to record the session, and so you can watch it again on our YouTube channel. So Claudio, thanks very much again for joining us, and the stage is yours.
Thank you, Professor Morland. Thank you very much. And I'm happy to be with you today, speaking about the mechanism of blood purification in extracorporeal therapies. The topic may seem a little bit boring, but especially for young fellow, I think is a very important for knowing the basis of what's going on when we have any patient in extracorporeal therapy. Mass separation processes to remove solutes occur by barrier separation, as in case of dialysis or ultrafiltration, and by solid agent, as in the case of adsorption. Now, diffusion is one of the main mechanisms utilized in mass separation process by barrier, and you see that small molecules increase clearance significantly when you increase blood flow, while middle molecules remain fairly stable and plateau quite early. The reason for this is that the diffusion coefficient for molecules tend to decrease significantly as solid molecular weight increase. And in case of protein binding, electrical charges, or lipophilic structure, there might be further reduction in the diffusion coefficient. So we are well aware today of what are the variables involved: solid diffusion coefficient, solid temperature, membrane surface area, concentration gradient, and distance for diffusion, which in this case is represented by the thickness of the membrane. Now, for moving from diffusion to a different mechanism called convection, we need to know very well the kinetics of water transport across the membrane by ultrafiltration. As you know, ultrafiltration in biology is a process of separation of plasma water from plasma and blood, and the ultrafiltrate contain crystalloids, but not colloids nor cells. And based on the response in terms of ultrafiltration at the given transmembrane pressure gradient, which is operated by the typical Starling forces, we can identify membranes with low flux, medium flux, or high flux, depending exactly on the capacity to have more or less ultrafiltration in response to a given ultrafiltration, to a given transmembrane pressure. Now, ultrafiltration is the process that occurs across the membrane, but when ultrafiltrate carries with it some solutes by a solvent drug, we speak about convection. And convection depends on the amount of ultrafiltration and the so-called sieving coefficient, which is the relationship between the concentration of the solute in the ultrafiltrate versus the concentration in plasma water. Now, this sieving coefficient depends very much on the sieve curve of the membrane, which characterize, as you can see here, high flux membrane, low flux, high cutoff, high retention onset, etc. This is good to remove middle molecules. However, in some cases, we must be aware that it's not just the molecular weight that makes the characteristic transport, but also the molecular radius, depending on the structure of the molecule. When the molecule is with oblong shape, then we have to look at the radius of the molecule rather than the molecular weight itself. Now, these two mechanisms are mostly utilized in dialysis, where we have a countercurrent dialysis to blood flow, maintaining the maximum concentration gradient. high flux dialysis, where we have some kind of ultrafiltration partly masked by amount of backfiltration inside the dialyzer. Hemofiltration, which is a purely convective therapy, where you have the ultrafiltrate production and reinfusion in the venous line. And finally, hemodiafiltration, where we combine basically diffusion and convection. Now, how do we classify membranes? We classify by composition and structure, biomaterials, the process, etc. We characterize performance characteristics like permeability, efficiency, flux, cutoff, adsorption capacity, and by surface modification, whether membranes are functionalized or not. Initially, we had membranes that were cellulose-based. Their wall thickness was very thin, but however, they were extremely poorly permeable and poorly biocompatible, with very low characteristic receiving properties. So they were prevalently used in diffusion. Subsequently, synthetic polymeric asymmetric membrane were mostly hydrophobic, and they were mostly utilized in case of convective therapy. In the meantime, specific synthetic polymeric microporous membranes were developed with high hydraulic permeability, high sieving properties, good hemocompatibility, and the possibility to combine diffusion and convection because of a reduced wall thickness. So how do you characterize a membrane? You characterize by this domain, which is pore density, number of pores per surface area, and pore size distribution. Pore density affects the clearance for small molecules, the membrane permeability coefficient to diffusion, and ultrafiltration. While pore size distribution, again, characterizes mostly the sieving coefficient and the sieving properties of the membrane. So increasing membrane pore density means increasing possibility to remove small molecules. And in fact, you see for a given blood flow, a membrane with higher pore density provides higher clearance for urea, for example. However, when you want to remove middle molecules, you have to move the pore size distribution towards a larger spectrum. And in this case, we may end up with high flux membrane with the same level of pore density, but larger pore size distribution. What is the distribution characterized for? It can be precisely controlled to avoid loss of albumin. We have to maintain high sieving coefficient for different molecules, but we have to avoid the loss of albumin. So, for example, membrane with high cutoff, where you expand the pore size distribution, the final cutoff may end beyond the retention of albumin point, leading to some albumin loss. That's why they can be used only in few sessions and not in chronic patients or in repeated sessions. Now, again, we said that we need to retain albumin, and therefore, the effort recently has been to move to the right the mean level of the pore size distribution, maintaining the same pore density, achieving the so-called mid-flux membrane or medium cutoff or high retention onset. What does mean this term? This term is characterizing a membrane with a similar cutoff where sieving coefficient is 0.1 or less for a given solute, in this case for albumin. And however, the retention onset, which is the point in which the sieving curve starts to be 0.9, is moved strongly to the right of the curve. So you see a much steeper curve that characterizes membranes that we define as high retention onset membranes. And here you see a selection of membranes depending on the different porosity, and it's like a catalog that the clinician can choose depending on what membrane and what type of solutes is targeted to be removed. We can say that membrane with thin low flux characteristics are good for diffusion of small molecules, certainly not diffusion of large molecules, certainly not convection, because as you can see here, the sieving coefficient is too low. Thick high flux membranes can be penalized for diffusion because of the thickness. They can work perfectly in convection, but finally, the optimized compromise is given by thin, high-flux, hydrophilic, hydrophobic membranes, where you can optimize both diffusion and convection for small and larger solids. What retains the solute and characterizes the sieving coefficient is a very thin skin layer at the blood membrane interface. And we know that the membrane behavior in vivo, in reality, may be very different from what we can measure experimentally for several different reasons. But one of the most important reasons is the interference of proteins at the blood membrane interface. That means that if you do high filtration rates, the membrane can be clogged, and your final sieving coefficient can be very different from the original sieving coefficient that was designed for the membrane. In this case, often pre-dilution is utilized. However, while the sieving coefficient increases, the overall amount of solute removed may be decreased. The other point is that the sieving coefficient of the membrane over time decreased significantly. That's why we always advocated a possibility to change the filter after 24 hours, even if the filter is perfectly working, because the sieving coefficient is reduced and the efficiency of the membrane is reduced. Another aspect is the hydrophilic hydrophobic surface. Here you see two examples of behavior of interaction of a drop of water in a hydrophilic hydrophobic structure of the membrane, where polarity of water may link with hydrogen bonds to the surface of the hydrophilic surface, while the chaotic orientation of water molecules have a kind of rejection effect at the surface of the hydrophobic membrane. This is of great importance because in case of hydrophilic surfaces of pores, you have a continuous fluid phase, and the fluid wet the surface of the membrane, leading to a continuity of the fluid phase in the pores, while there is a discontinuous fluid phase in pores of hydrophobic materials, where you only can use convection in order to remove solutes. Today, we have surface microdomains that may limit interaction with proteins and cells, strongly improving the capacity of the membrane to be biocompatible. Plus, we can control roughness, and the characteristic can be studied by scanning electron microscope. Also, we have an effect of possible surface modification that may affect biocompatibility, permeability, having non-fouling effect, gaining bioproperties to the membranes. So, for example, we have seen in the past that the membrane can be an active component that may remove endotoxin or pro-inflammatory mediators by adsorption. Today, we know that we have some membranes, in this case AN69, with high capacity for removal of beta-2 or other molecules by adsorption. And we have membranes in which the surface can be functionalized using polyethylenimine groups that may further increase endotoxin adsorption or heparin binding on the surface, giving specific bioproperties to the membrane. In this case, these membranes have demonstrated high capacity of removal of pro-inflammatory mediators, not by clearance through convection or diffusion, but simply by adsorption. And there are other membranes, for example, polymethyl methacrylate, that has high capacity for adsorption. And it is structured in a way that adsorption may become an important component. So on the past, while we try to improve hemocompatibility by modifying the surface, subsequently, we modified the porosity to change the flux and the sieving coefficient, and recently to add the bioproperties to create some kind of bioactive membranes. This is the case, for example, of membrane structure functionalization with alpha tocopherol. The surface of the membrane is functionalized with vitamin E that may lead to a specific action in terms of bioreactivity, control of protein oxidation, reduction of LDL oxidation, reduction of erythrocyte damage due to oxidant stress, and reactive oxidant species are deoxidized. In this case, the addition of vitamin E to the membrane somehow offer a further option to a very antioxidant therapy, which is preferable because it acts exactly at the point in which normally reactive oxygen species are generated. In this case, taking advantage of this membrane, we can provide a kind of antioxidant CRRT. And this is something we're studying right now. The preliminary experiment show a significant limitation in generation of reactive oxygen species compared to standard membrane without any vitamin E binding. So today, there is a multidimensional membrane evaluation that include material, the sieving, hydrophilic, hydrophobic compounds, retention onset, biocompatibility, permeability to water, electrical charges, cutoff, thickness, and permeability diffusion coefficient. But looking at the spectrum of retention molecule by proteomic assessment, we know that there are several solutes that accumulates in blood of patients with AKI, sepsis, and chronic kidney disease that cannot be removed by classic dialysis membranes. The reason for this is that these molecules are in the range of molecular weights that go from practically 15,000 to 50,000, and they are responsible of most of the toxicity, infertility, inflammation, anemia, cardiovascular toxicity, and inflammation in general. In this case, hemoabsorption may be the solution because overcomes the limitation of HD membranes, has a potential selectivity of the removal. You may place the sorbent in contact with blood, no alteration of anticoagulation regime, no problem of circuit pressure profile. But of course, we have requirements. We need demonstrated effectiveness, hemocompatibility, mechanical strength, no fouling or clotting activation, and well-designed cartridge. Now, the original sorbent materials like charcoal were very bioincompatible. Only with the beginning of this century, new resins have appeared on the scene with smooth surface and improved characteristic of biocompatibility. More recently, new neutroporous macroporous resins have been developed composed by styrene divinyl benzene copolymer that are characterized by very smooth surface and very high biocompatibility. We have studied by scanning electron microscope the morphological analysis of the surface and the pores up to a very, very high magnification to study the characteristic of these sorbents. What are the forces involved in adsorption? Van der Waals forces, ionic bonds, hydrophobic bonds. These are the three forces involved. And how do we characterize these sorbents that are mostly coming in beads, sphere, or fibers? Again, by pore density that describe the amount of adsorbate, and by the pore size distribution, which describe the molecular spectrum in the adsorbate. We have to point out that 1 gram of this substance has a surface area of approximately 1,000 square meters. So there is enormous surface for adsorption. And in fact, once you measure the isotherms, which are basically the clearance curve by adsorption, you can see that the single molecule is affected in the characteristic of the isotherm by the pore density. Number of pores and surface area describe the capacity to absorb a specific molecule, and the isotherm at equilibrium describe the maximum amount of solute absorbed by a given quantity of the sorbent. While when you speak about different pore size distribution, different solutes present different isotherms that are characterized by the sieving properties of the pores. Today, thanks to nanoscale molecular sieve control technology, manufacturers can adjust the pore size distribution according to target toxin and molecular weight and radius of the molecules that need to be removed. So again, we see that we have microporous, we have mesoporous, and we have macroporous membrane, more or less with the same concept of the pores that we had elucidated before for dialysis membranes. And this has an impact on the target molecules. As you can see here, large molecules and essential proteins can mostly be removed by meso or macroporous structure, while microporous structure limit their action up to 15 to 20,000 volts. Hemocompatibility has been studied in our laboratory for these new sorbent materials, and both in static and dynamic tests, viability, apoptosis, and necrosis of monocytes have demonstrated no difference compared to the medium. This is in part due to the external and surface coating that describe a very smooth and easy interparticle, extraparticle, and intraparticle flow of the fluid phase. We know that in this case, the external but surface coating of the material is important. So not only at the surface of the membrane, there is a kind of a coating, but also inside the pores, allowing the maximum biocompatibility. Otherwise, in the past, the coating at the external part of the bead impeded the utilization, exploiting the maximum capacity for adsorption, because adsorption was only limited to the external part of the sorbent. Today, this type of coating allows small, middle, and large molecules to enter the structure of the pores, and therefore we can demonstrate the capacity of adsorption for an enormous amount of square meters and surface area. We studied, for example, the mass transfer zone together with the isotherms, and this described the curve and the distance between maximum utilization and zero utilization inside the cartridge, because we wanted to analyze specifically the different variables that may affect the flow distribution, including particle diameters, packing density, interparticle porosity, path tortuosity, length, diameter, fluid viscosity, etc. We studied the flow distribution with a CT scan, and we demonstrated a very homogeneous flow distribution in the cartridges, showing maximum utilization of the available sorbent. More than that, we studied the so-called mass transfer zone that I just explained with a specific prototype designed by us with areas for specific drawing that allow us to study concentration along the length of the cartridge. And the experimental mass transfer zone displayed a very, very short distance, showing that the maximum utilization of the sorbent inside the cartridge is achieved. We have example of functionalized sorbents, for example, polystyrenic fibers with PMX or cross-linked or DIA. And we want to suggest, as we did in the past, a kind of utilization of adsorption in sepsis based on the time frame of the different events and cascading sepsis. When you have infection, you may have a pathogen and endotoxin circulating in blood. At that point, you have Seraph, polymyxin B, Alteco, LPS sorbent, or more recent sorbents that may remove endotoxin. But when you have cytokines, pumps and dumps in the phase of immunodysregulation, then in this case, you may need adsorption of these molecules by specific columns. And then when organ damage occurs, failing organ can be replaced by different CRRT techniques. But the main issue is to try today to prevent the progression of sepsis toward organ failure. Seraph is a column that somehow reproduce the heparan sulfate at the inner side of the endothelium that has shown high capacity to remove pathogens, bacteria, and viral particles. Polymyxin B-based medical devices like toromyxin have shown high capacity to remove endotoxin and finally, cytokine adsorption can be achieved using the new resins with the characteristic of neutral macroporous structure. Studying in vitro the capacity of this sorbent, we have detected high capacity for removal of pro and anti-inflammatory mediators already in the range of 30 to 60 minutes with high capacity, therefore, to maximize the response and achieve the concept of peak concentration hypothesis, which is a normalization of the peaks in different pro and anti-inflammatory mediators and somehow restoring a kind of immunohomeostasis. How do we apply our cartridges? In direct hemoabsorption, in hemoabsorption plus hemodialysis or CRRT, in plasma filtration adsorption or plasma filtration adsorption plus CRRT. And in case of DPMAS, this means double plasma molecular adsorption system, where the plasma separated from blood is circulated through two different types of sorbents to achieve liver support by removal of bile and bile acids and also pro-inflammatory mediators. Putting the cartridge before the filter is not a problem from a technical point of view. It's quite easy. The only thing we have to be aware of is that any type of cartridge saturation occurs between 6 and 12 hours. So it makes no sense to use after 6 to 8 hours the cartridge because it is already saturated and you will expose the risk for coagulation without additional benefits. We also need to know that adsorption occurs also for beneficial substances, including, for example, antibiotics. And this is a very important thing to know and to consider. Recently, we did a very interesting experiment where we functionalized the surface of the sorbent by vancomycin. And by using this functionalized sorbent, we were able to show a significant bactericidal effect in vitro, showing that probably we can use functionalized sorbent with specific drugs to bring the drug to the patient without having systemic effects. So in the evolution of sorbents, we had an aspecific sorbent that originally was problematic. Now we have an aspecific sorbent, which is probably optimized, and the fact that it is non-specific provides restoring immunohomeostasis because remove peaks of both pro and anti-inflammatory mediators. And in the future, we might have specific sorbents that may actually be characterized by some kind of specific functionalization, what we call the smart sorbent. We have recently provided a book on this concept and also published the results of the ADKI consensus conference. We prefer to use the terms today hemoabsorption rather than hemoperfusion because really this is the term that probably clarify more the characteristic of this therapy. So we can say that moving from the original CAVH and then CAVHD, we moved to CVVH, CVVHD, and then hemodiafiltration with the plasma. Then we had third generation and fourth generation of CRRT machines, including the possibility to support multiple organs and provide extracorporeal organ support. And today, we have probably the new horizon, which is characterized by the hemoabsorption. Where are we today? We are where CRRT was 30 years ago, and we need to structure research. We need to identify patients and the phenotypes that are likely to benefit from hemoperfusion and hemoabsorption. We need to establish adequate dose, frequency, and criteria for application. And we need to identify target molecules and biomarkers to do biomonitoring. We need to identify adequate endpoints for clinical trials to establish evidence. And endpoints can be biochemical, biological, physiological, pathological, and finally, survival will be the ultimate. We need to consider potential side effects and contraindication to promote a medical alliance with industry for progress, and we need homogeneous terminology. Now, I take the liberty to invite you all to the Vicenza course, June 11 to 13, at the Vicenza Convention Center. We will be speaking about AKI, CRRT, extracorporeal organ support, and in general, critical care nephrology. This is the 42nd edition. We have an outstanding faculty, and we're really waiting for you in Vicenza to further elucidate some concepts and possibly share some ideas for new studies in the future. With this, I thank you very much for your attention, and I return the line to the organizers.
Thank you very much, Claudio. We have several guests online, and we have a small crew here in the room. And may I ask anybody has any questions? While you're thinking, it was fascinating at the beginning of your talk how you led the way of thought from the filters, the membranes, to the adsorption techniques. And from your talk to me, it became obvious, and correct me if I'm wrong, that these kind of technologies just fill in a gap, an unmet need in the story of hemoperfusion. So it's not like a new therapy. It is perfectly in the evolution of the story, and these can complement each other, as you nicely depicted at the end of your talk.
Yeah, I think that the story of uremic toxin and toxicity of specific molecules is progressively unfolding. And the day we know that, for example, beta-2 microglobulin accumulation or specific molecule accumulation correlates with specific outcomes, removing those molecules become like the target therapy. It is interesting, however, that in some cases like sepsis, we have for cytokines a typical jump in the response of innate immunity and subsequently of the adaptive immunity. So we have up and down of peaks of pro-inflammatory mediators, but also anti-inflammatory mediators, because some patients have the monocytes that are completely paralyzed. Now, we have studied the behavior of monocytes after the hemoabsorption. And we found, for example, that monocytes restore the capacity to express on the surface HLA-DR, which means restore the capacity to present the antigen, which is a kind of reduction or elimination of the immunodysregulation, which characterized the septic syndrome. And we're not really curing the sepsis. We're simply giving the patient the weapons and the situation to be able to restore the immune response.
Thank you. I encourage those who are online with us to put in questions in the Q&A, pressing the Q&A button at the bottom of your screen. While we are waiting for other questions, I mean, you mainly mentioned in the adsorption story the Jaffron technology, but most of the studies are published with cytosolve. And may I ask you, if a junior approaches you, that which adsorber should he or she use in a patient with septic shock, vasoplegic shock, whatever, then so which one would you recommend? Or are you aware of any data that shows that one is better than the other? Or is there a pathophysiological rationale that one bead is better than the other bead?
I think that basically, I was interested in adsorption since the last 30 years. And I was the one doing the first experiment with the sorbent called beta-sorb, which was the grandfather of the cytosorb of today. I became aware recently of other adsorption materials, and we moved to experiment and do studies on the polymyxin B coated fibers for adsorption of endotoxin. And more recently, especially during COVID, we became aware about this new technology by Jaffron that is characterized by macroporous structure and some specific coating characteristics, which makes very biocompatible. Now, it's not to me to say what is better. And in fact, in the past, you may remember, I did several studies on membranes. I never mentioned that one membrane is better than another. I personally think that the process of adsorption is of extreme interest and should be studied, should be promoted, should be used because, as you said, it complements what we can do with other membranes. Now, the commercial issues are not of my interest. I can tell you that basically the characteristic of the sorbents are quite similar. It might be a matter of a little different sieving properties, biocompatibility properties, or even the quantity. For example, in the Jaffron cartridge, the quantity of the sorbent is more than in the cytosorbent. But at the same time, they have pediatric cartridges, for example. So it depends very much. I think that one should use the adsorption technique, should probably use all available sorbents in order to create his own experience and then decide what can be used. There is another issue, which is in some hospitals, we are not deciding what products we buy, but simply there is a kind of administrative process that leads to the acquisition of the materials. So I'm not able to say that. But I can tell you that in some cases we studied specific characteristics. We have done now several experiments for removal of bilirubin, for removal of myoglobin, for removal of other substances. Simply, I had the chance to do my first experiments with CytoSorb many years ago, and now I'm doing experiments with this more recent adsorption device. I personally think that the adsorption concept should be promoted rather than promoting one specific device. Thing that I don't want to do.
There is a question, Claudio. If you click the Q&A from one of our colleagues, Luca, she's. Shall I read it out, or can you see it?
Chat.
Anyway, not the chat, it's a Q&A. But do you think the new filter technology options will aid precision CRRT? Yeah. So
do you
it's
think
about new filter
technology option will aid precision CRRT prescription? What diagnostic do decide what filter? Okay, so this is a perfect question that describes the logic approach of a mechanistic therapy, that you have one molecule, you want to target the molecule, you need a specific therapy. The problem is that we as doctors do not have the target molecules yet. We have not identified specific target molecules. We are starting right now. We're not even able to monitor with bedside test cytokines or other molecules. So engineers often tell me, okay, I can do whatever you want. Tell me what you want. And the answer is, well, I don't really know what I want. Let me make an example. In chronic dialysis patients, we use urea as a marker molecule. But urea is not toxic at all. It's a good biomarker because its accumulation describes how much dialysis I need. Its removal describes how much good job I'm doing with dialysis. And I know very well the distribution volume and the generation. So I know everything about that. For many other molecules, we are just starting the information of this. So it's something that we have to research. That's why today, for the moment, a kind of non-specific adsorption to me is much better, like a protecting umbrella, rather than being specific for one molecule that may represent the magic bullet, but then we have many other molecules that affect the syndrome. I can tell you that this is a kind of pragmatic approach, if you want, but the other concept is that the results we see from the biological point of view, from monocyte point of view, from the hemodynamic response is very interesting. So we know that we are doing something good. We have just to understand better what we are doing. And that's why I'm trying to put together many companies that do sorbent with companies that do biomarker, because we would need a bedside monitoring of what we're doing. And at the end, we will be able possibly to come up with precision CRRT prescription.
You have another question in the Q&A, if you could.
Yes.
It's a bit
One
long.
concern I had in relation to absorption is that production of such molecules as the shock is such a rate that as soon as the absorbent is removed, pretreatment levels are reached within hours. Do you believe this is a valid concern? Okay. The kinetics of cytokine, as the colleague has pointed out, are very complex, and the cytokines have a very short half-life. So you measure cytokines throughout the therapy, and you find cytokines going up, down, up, down. But there is a kind of an observation that removing cytokine from the circulation may lead to a progressive withdrawing of molecules from the interstitium, so that there might be a kind of modification of the peaks at the blood level, maintaining the high concentration in the point where the specific origin of the sepsis is. And this may lead, indeed, to the possibility to identify a specific source of the sepsis. It's being called the cytokinetic theory for sepsis. The other thing is that we don't know exactly what we are doing, but certainly something we are doing, because 3 hours, 4 hours after the application of hemoabsorption, immediately we see that there is no need anymore for noradrenaline, and there is an improvement in hemodynamics. So we're doing something good. The response of the monocytes start to be there again. We are doing something good, but we're not there yet in terms of having a complete mechanistic explanation of what we're doing.
This is very good, because we've got a lot of work to do, right?
A lot of work.
A lot of research.
But you see, this is a special call for action, because many people just say, oh, there is no evidence, and then they stop there. I think we have to generate the evidence. So we have to do our studies. We have to plan very well-designed trials. And also, the other thing is, we have to be careful on how we design the trial. We had recently an ADCI meeting on endpoints for clinical trials. We have pointed out that we need to identify the typical endophenotype of the patient that is kind of homogeneous target for the therapy. Otherwise, you may end up with a neutral, positive, or negative response, and then you don't know what to do with the therapy, and then you may drop the therapy, throwing away the baby with the baby wash. So the problem, I think, is we have to clarify, identify the patient, identify endpoints that not necessarily are mortality at 90 days today. They might be so in 2 or 3 years from now, but not now. Now we need to identify specific action and try to concentrate on the ideal dose and frequency. I remind you that it took us approximately 30 years to decide how much CRRT to give to deliver an adequate therapy. Before our paper in Lancet in the 2000, There was no agreement, or nobody even was interested in dose of therapy. And it took 30 years to come there. So we just started with hemoabsorption. I hope that it will not take 30 years to go there.
Thank you very much, Claudio. Our time is up. So finally, I would like to thank especially you for joining us, and for the audience, mainly from the Department of Anesthesia and Intensive Therapy at the University of Semmelweis. And of course, without the support of the translational medicine, this meeting could have not taken place. So safe journey to you wherever you go, China, or you just returned from China?
Just returned from China and from Argentina. But let me tell you something about the University Semmelweis. I was always a great admirer of Semmelweis. And when I received the first call, when we started using the Carpe Diem machine for the first time in the first neonate, I was in the Semmelweis University.
All right. Well, it gives a special flavor of this meeting. So thanks again. Thanks, everyone. You will be able to watch the YouTube video of this meeting. Last time, the last recording was watched by almost 150 people. So let's hope that we will increase that number over time. And we will see you in 2 weeks' time, and we'll talk about that lactic acidosis is fake. And the lecture will be given by Robert Roberts from Australia, who will get up at 2 a.m. to give us his lecture. So, Claudio, thanks again. Everybody else, have a nice evening. Bye-bye.
Send me the link.
We will.